Today I'd like to talk about two new tools that we recently developed. One is called LitSuggest and the other one TeamTat. Before I go into details, I'm going to show you a general pipeline, how text mining is used for knowledge discovery. Obviously, this is not the only way, but this does represent a lot of research in our community. So let's say a user have a need for a specific topic. So they were trying to retrieve a subset of articles that are relevant to that topic. And once they have that set of documents retrieved, they can develop tools to recognize concepts that are important for biological research, such as genes, diseases, and drugs and chemicals. And one step further is to map these mentions of disease mentions or gene mentions into standardized or unifier code. And this is important because even within the same paper or across papers, the same concept may be expressed in different ways. Now, once that has been analyzed, one can try to analyze the relationship between these extracted entities, and that generates sort of a relationship or entity graph, which can be integrated later on with human domain knowledge for further knowledge synthesis or discovery or generating new hypotheses. So at NCBI NLM, we have been developing tools along these lines. And for tech savvy or text mining savvy users, you'll probably find the most inner circle, the yellow color data sets to be most useful because you can develop your own algorithm based on these corpus. For those that are bioinformaticians, you have programming skills and you would like to use softwares on your own problems, you can sort of find our tools developed in the middle circle, where these are open access, open source software tools. And we also make the outlayer of the tools that is directly user facing. These tools are intended for users that are not NLP specialists, but they would like to try NLP, or they like to using that just for application of NLP for their domain problems. And so this does not require programming skills at all. All you need is just a web-based access. So in other words, if you have a web browser, you can using these tools. And one of the probably best known tools from our group is called Tator, which is a database of annotations of 6 different entity types that include gene, disease, chemical, cell line, mutation, and species. So we developed these tools, state-of-the-art tools for tagging those entities in the articles. And we go one step further in Tator that we apply these tools to the entire set of PubMed articles, which is over 30 million now, and the entire set of open access PMC full-length articles, which is over 3 million now. And we generate and store these annotations in a database and then display them to our users at the web front, as you can see here on the right. In total, we have over 1 billion annotations now, and that number continues to grow because we analyze, we do nightly update every day for new papers coming into PubMed or PubMed Central. And this work is carried out by 3 very hardworking and talented fellows, researchers in my group. And Tator, one of the most popular uses of Tator is through its programming interface. So this was made available in around 2015. And since then, we have had over 600 million requests from all over the world. They use, and with different use cases, such as gene prioritization, such as knowledge-based, literature-based knowledge discovery, or downstream text mining applications. For us, we use Tator as well. And we use them extensively with, in a couple of years ago, with database, with biological database curation to support biocuration. And to this audience, I think I don't need to stress the importance of those biological databases, which play a key role in today's biomedical and bioinformatics research. But data curation is very costly, and because it's done mainly by human manually. So we're working with these different database separately, but we find together with the findings with different databases that NLP can really scale up the curation process by improving the efficiency without sacrificing in the data quality. And we published a couple of papers, our experience with these different databases. And after these papers were published, there are actually more databases, individual databases reached out to us and asking us for similar help. And then at that point, we find we actually have limited resources to working all different databases. But we looked at their needs. We find out a common theme is that they really want to scaling up the literature triage process. In other words, they want to find a set of papers that are relevant to the topic that their database is curating. Okay, and their status quo they're doing now is they usually have a set of predefined or hedged PubMed queries. They search PubMed, they get the list of list of results back, and they download the PMIDs in a spreadsheet, Excel sheet, and then they open up another window of PubMed, and then they enter the PMID, and they look at individual articles, and then they record a yes or no, relevant or not relevant, going back to their spreadsheet. Okay, so this is, as you see, this is not very efficient. And what their desire or what they would wish for is tools that can be more accurate than keyword-based searches in PubMed. They would also appreciate tools that can do regular scan of the new articles on a regular basis, either daily or weekly. Typically, the database curation is comprised with curators more than one person, so they like this process to be collaborative, and they like this system, the results of the curation of the triage process to be readily available and integrated, readily available, and can be imported to their curation pipeline. And that's the purpose that we developed this system called LitSuggest, which is a web-based system for literature recommendation and curation. And all you need as input for LitSuggest is just a set of positive articles. And if you also have a set of articles that you have reviewed but rejected for relevance, that can serve as negative examples, but that's not required because we can randomly generate a set of negative articles. And then we put them together into an ensemble learning using different advanced classification or NLP techniques. And then we build a classification model so that when new articles come in, they can be automatically scored into positive and negative set. Now we add a visualization, which is very helpful, welcomed by our users, so that we can see the new articles, what are the ratio or proportion of positive and negative in different beings according to their confidence score of the prediction. And then we can also visualize the salient word or informative words in the positive set, which itself is very important and useful just for example, for illustration purposes or for using for a presentation. And down under the visualization, you see the interface where users can actually annotate and curate, and then they can easily download and share with others. So we use one of the reasons we developed LitSuggest is because we really had a need for that. Also for ourselves, when we developing LitCovid, which is a curated literature hub for COVID-19 papers. And this is very important, especially at the beginning of the pandemic, because COVID-19, this particular term, was not used in the scientific literature, even though there are many papers describing COVID-19, but there was no controlled vocabulary for COVID-19. So there's just many different ways COVID-19 is expressed in the literature. So as you can see, using a PubMed query is not going to satisfy or retrieving both high precision and high recall. And that's why the machine learning classification really helps. And of course, we're using this for this purpose, but this really goes beyond a single topic. Any topic that you're interested in, as long as you have a set of relevant articles that you have reviewed, you can use LitSuggest, and it will find papers relevant for you later on. And others are using that as well. For example, Rhea database in Switzerland, this is a database of chemical reactions. They're using LitSuggest extensively for their literature curation purposes. They do weekly scan for new papers. They also scan the the legacy papers that already existed in PubMed, and they find over 500,000 papers that could possibly relevant for their database curations. So at this point, I'd like to switch gear. And now let's say you have a set of papers and you want to do further analysis. And you probably want to do concept recognition and relation extraction. And you may have tools like PubTator to help you to get started, but you may also have concept that you or a relationship that you want to extract that are not already available by feasible by existing tools. So in that case, you probably want to develop something on your own. But I can tell you is that, and this is also alluded by the previous keynote speakers, that the current method is not very generalizable to, if you develop something for protein-protein interaction, it's probably not going to work that as same level if you apply to drug-drug interaction. So, but we do know that AI and machine learning methods work, but they require annotation data for training. And so for that purpose, you do have to have humans to build corpus or training data for machine learning AI methods. And this is still costly and very tedious. And we're not going to save the manual curation process, but we're trying to make this process as smooth as possible. And that's why when we have annotation project ourselves, we were looking for tools that can really do this. And we were aware a bunch of tools that are available for text annotation, but in the end, we developed our own because we really want a tool that is web-based, and also free to use, and they can be seamlessly integrated with PubMed and PMC articles. That's our main focus, and it can using machine pre-tagging. And also one thing that we find is lacking in almost all existing tools that we want a tool that can be used for managing team annotations. So this slide is prepared by Rizata from my group, and as you can see here, that there are two rounds of annotation, usually starting with independent annotation and then starting with the collaborate annotation for generating a text corpus. So this is a very short animation about how the interface of our tool. So for example, here we want to annotate a chemical. So we just highlight the chemical name in the title, and then we can enter a concept ID. In this case, we choose the mesh ID for this particular concept for normalization. We can click the box for annotate every instance of the same instance, a same concept. See, and after that, basically we annotated the chemical names in this abstract, in this PubMed article. And that's essentially the individual annotation round would include. And then the next thing is really the key is for a project manager really to go into the collaborate annotation round where we can produce and visualize the annotation agreement. We can look at the differences between the annotators, and then the two annotators can work one-on-one about resolving the differences and reach consensus. And here is another animation to do that. So for example, in this assignment, we have four annotators with a project manager. Here we have 4 articles, for example, and then once each article has 2 annotations, then we can close this individual annotation round, and then that would bring us to the process of looking at the inter-annotator agreement and visual differences. So for example, in this table, we can directly click on the statistics, and then once I do that, we will see different statistics. And here we mainly show you the 2 things. One is called a full agreement. Full agreement is for knowledge for concept annotation, where we require both the text span and the concept ID to be fully agreed by 2 annotators. So you can see here, for example, articles we have, for these 4 articles, we have between 80% agreement to 98. And then we also have a single annotation. So these are the annotations made by 1 person. So you can see we have less than, a single digits percentage of the annotation are made by just 1 annotator. And then we can look at details about these differences and agreed annotations. So everything that are highlighted in green are the annotations. And then we can also see that there are underneath lines here, and some are colored in black. which means that in this particular case, NCK, for example, the annotations are different. And also the ones that are underlined by gray line, this says that these annotations agreed with each other perfectly, both the text span and the concept. In the black, as I mentioned, this would be different annotations, even though they both highlighted NCK, but they gave a different gene IDs in this case. So this one is very clear to see there's differences. And P155 without underline, which just basically showed this was only annotated by one person. Okay, so this was very easy to see visually. And then after that, basically we can now assign to the two annotators to engage in the collaborative annotation round. So this is basically how our tool is used. As you can see, you do all the annotations in one place, and you can from the beginning, very beginning to the very end. So we use this, as I mentioned, because we had a two annotation project. One is to annotating 550 PubMed articles for gene mentions, and the other one is for annotating 150 full-length articles for PMC full-length articles. And these are finished, and they also the results of these two corporate are also made public available. Obviously, we focused on English language, but this tool can support other languages as well. And we know our users are using different languages like Portuguese and German. We focused on medical literature, but this tool can accommodate other types of documents as well. So our users, for example, are using TeamTat to annotate documents in clinical documents and doctor notes, because one thing nice about TeamTat is, in addition to the web form, you can also download and install this tool locally, which is very important because if you have sensitive and private data you don't want to upload to server, you can do that on your own locally. And even though we aim this tool for the biomedical domain, but obviously this tool can be applied outside that area as well. So in summary, that I showed you two different tools today, in sort of along with other tools that we have developed over the years from our group. And I'll be happy to take questions now, or you can send me emails. And also a shout out that we have openings for postdoc positions. So if you or you know someone who might be interested, please feel free to contact me, or looking at more details, job descriptions with the link below. Thank you.
Thank you, Zhiyong. So I'm going to move quickly because we have 2 minutes. So I want to have some questions answered. So there's one question that says, what are the performance scores for LitSuggest? It would be nice to avoid too many false positives. Sorry if I missed that in your presentation. That's why look at code those.
Not at all. This is a great question. The detailed, there is no, so what I'm trying to say is there's no one number for the performance. It really depends on the task, annotation task. So if you have training data that is large enough for text mining for classification, then you can get a very high good performance. In some of our cases, I can give the example. For example, for when we're looking for COVID-19 relevant papers, we have F measure above 90%. And similarly for the Rhea database, like the chemical reaction database, they're finding those papers. So it really, there is no one answer. It really depends on your training data. But if you want to look more, you should looking for our paper on the LitSuggest. We have some examples, and we have also performance on those tasks.
Thank you. So last very quick question. So you use all NCBI vocabulary, right, for the annotations. Is it possible to adapt that to any other databases? Like, for example, you mentioned it's work with Rhea or UniProt. Is that possible to link to UniProt IDs instead of gene IDs, or both?
Yeah. So we try to using community standards, and when there are more than one standards, we just using one that is best fit with our text mining purposes. There are along the lines of these chemical entity types, for example, cell lines, we don't use NCBI cell line ontology. There's not such a thing available, so using something else. And when you, for example, for the genes, we use genes, but I believe that there's a conversion between gene IDs and source code IDs. So person certainly can do that.
Okay, thank you. All right. So now, okay, there's another question. Maybe we can answer it very, very quickly. So is the model in LitSuggest recalibrated or retrained from the user feedback as they explore the article results, labeling them relevant or irrelevant?
Yeah, exactly. That's exactly what LitSuggest could do. If you can curate both relevant and irrelevant papers can be used for both relevant and irrelevant could be used for the next round of annotation. So your classification model can be calibrated as you do more rounds of annotations or classifications. Yeah, exactly. That's what is a feature of LitSuggest.